Good afternoon. We're going to start the session now. It's my pleasure to introduce Dr. Gary Peltz. He's a professor of anesthesiology at Stanford University. And he's going to talk today about the genome engineering from safer drugs to liver regeneration. Thank you. Dr. Peltz.
I'll use this one. Thanks. Turn that one off, though, so it doesn't cause things. Can I have the lights down a little bit, because I'm going to show a lot of histology slides? Hi. Can you hear me okay? Pick it up. Is that better? Okay. I hear just echo up here. It's kind of funny. Today, I'm not going to talk about mouse genetics, but you'll see there'll be mice involved. What I'm going to do is talk about how we've humanized the liver in mice, how we made that model and why we wanted to do it, and then how we've modeled certain genetic diseases, and lastly, how we've done predictive toxicology, and then how we'll move that into human liver regeneration. I think for an anesthetic pharmacology audience, I don't need to introduce the problem at any length, but just to say that the basis for testing the safety of drugs is done in animals, primarily rodents. And you know, humans and animals are very different in the way they metabolize and respond to drugs. And that's why 40% of the toxicities that occur in humans are not predicted at all by animal models. So we decided to do something about that a few years ago, and this work was done in collaboration with the Central Institute for Experimental Animals in Tokyo, Japan, and we decided to build a better mouse. And what we did was we took an immunocompromised mouse, we introduced a transgene, the thymidine kinase transgene, and it's only expressed in the liver. And the reason we did that, the mouse is called the TK-NOG mouse because of the thymidine kinase transgene, but what you can do is just give the mouse 2 doses of ganciclovir 7 and 5 days before you transplant human liver cells in, and what you end up with is a mouse with a humanized liver. So why do this? There were other models of liver humanization available, but at the time I didn't like them. And it's because they either expressed a very nasty transgene, uroplasminogen activator, or had knockouts that created the damage in the liver, which allowed the human cells to replace. They had ongoing liver damage, and if you were going to study drug toxicity, it wasn't great to have this continuous liver toxicity. So in our model, the toxicity is temporally controlled. We just give 2 doses of a drug. We wipe out the mouse's liver and put the human liver in. Because of that, the mice are otherwise okay, and they can survive a very, very long time. And just to show you a little bit of what you can do with it, so again, we give 2 doses of ganciclovir, put the human cells in, and we measure in the blood peripheral how much human albumin they make. And just to show you, here's a typical experiment. We do the transplantation. About 8 weeks later, you can see the human albumin there, and this is an 8-month experiment. They maintain their weight, and they maintain their human biosynthetic capacity. So you've got a stable liver that you can then do experiments with. So we make a high level of human reconstitution. Those of you who are interested in these things, we do get zonal expression of glutamine synthetase. For those of you who don't know what I'm talking about, don't worry, but we do get mature liver that recapitulates what goes on in humans. And what I'll do is just summarize some of the early work we did, which was just to ask, could we predict human metabolites? And what we did was we took a drug in development. It was a drug called clemizole. We worked out the pathways. They were very different in rodent and in humans, mapped the pathway and showed by going into human volunteers that the metabolites we predicted in the humanized mouse were indeed present in humans. We could even model a drug-drug interaction in the mice and so on. So I encourage you to look at our, if you're interested in this area, you can look at our JPET paper. But then we went on, and it's something that didn't get a lot of attention, but I kind of liked the results. The question was, could we do human pharmacogenetics in these mice. And the idea, the concept was, and I'll just use the data here from CYP2C19, could we give hepatocytes from humans with different genotypes? In CYP2C19, there's normal, there's deleted, and then there's highly active ones. And the concept was, could we put the different ones in and see a different rate of generation of the human-specific metabolite, which if we gave mefenatine, you got 4-hydroxymefenatine. That was a human-specific metabolite. We also worked with diclofenac, and there's a few other drugs in the paper, but the results were really quite striking in that if you looked at the rate of generation of the human metabolite, this is the average of the wild type, this is the one with one impaired gene, and these were the hyper people. You could distinguish them. And in work done by Ming Zhang, who's the biostatistician, where we looked at the variability, with as few as less than 10 donors, you could perform human pharmacogenetic analysis. And remember, in these mice, you're controlling the diet, there's no confounding medications, et cetera, et cetera. So the data is really, really clean. So it was a nice way to do human pharmacogenetics. And initially, I was going to do a lot more work in this area, but we decided to pursue a different problem. And that was, could we predict liver toxicity? The liver is the main organ affected by drug toxicities. And so I picked sort of the big mother of toxic agents called phyluridine, or FIAU. It was a drug in 1992 made by Lilly, very, very potent against hepatitis B virus. It was tested in rats, dogs, and even in primates, gave no evidence of toxicity. Lilly was pretty comfortable with it. They were pretty much booking revenue on it. Unfortunately, when it went into clinical trials, a very bad thing happened. They gave it to 15 patients, 7 of them died. It was the worst disaster in drug development in North America that we had. The National Academy of Science commissioned a retrospective analysis. Did they hide anything? Did they miss anything? They looked over all the animal data and concluded there was nothing missing or hidden. They even did post hoc testing to see if doing other things could possibly reveal the toxicity. There was no indication. So it was a real problem. So what we did was, and just to show you, this is what happened in the patients. Everything looked good for about 8-9 weeks. And then somewhere around week 9 and 10, a severe liver failure developed, which was a bit unusual because it had lactic acidosis. So it was later in onset, and it was accompanied by a lactic acidosis. Well, we gave mice, and in this case, it was a very high dose, about 1,000-fold above the human dose, but this was a dose that didn't bother rats in a 6-month study. We gave the dose to a conventional mouse, and sure enough, they looked fine. But we started the dosing on Monday. On Wednesday, I was called by the veterinarians, Dr. Peltz, what are you doing to your mice? You can't continue these experiments, because by Wednesday, the mouse looked awful. You could just tell it was very sick. And when we did the testing, you could see the elevation in the liver enzymes. I show the individual data points. This is every mouse that we treated. You can also see the elevation in the lactate. So we got the same clinical presentation in the humanized liver mice as occurred in the humanized mice. And if you look in our paper, you'll see it's dose dependent. As we lowered the dose, they could survive for longer treatment periods and so on. But what's really interesting is this is a human liver. And for those of you who are used to looking at liver, it's just sheets of cells. I don't know if the light's down enough, but you see all the holes, the Swiss cheese? Those turn out to be fat droplets. And the other thing that's interesting, let's see if I'll show it more. So here's our humanized mouse liver, and you can see visually where the mouse is, and you can see it with a human antibody. And when you look at the tissue right in the humanized area, you can see we're getting the same pathology as occurred in the humans. Those are fat droplets. We've done a lot of staining. And what's really interesting is this is a normal liver, and then these are a blow-up of the area. These are the mitochondria. And I don't know if you can appreciate it, but there's railroad tracks across the mitochondria called cristae. Well, if you look in our FIAU-treated mice, you can see all the fat droplets. Those are why the thing looks like Swiss cheese. This here is a blow-up. These are the mitochondria. They're all misshapen, the cristae are gone, and so on. So ultrastructurally, we could recreate the lesions that occurred in the patients in the mice with the humanized liver. And so it was a nice example of, had this technology been available 20 plus years ago, you could have avoided that kind of a toxicity and that kind of a disaster, actually. So then we went on with a second drug with a more subtle toxicity. Bosantin is an agent used to treat people with pulmonary artery hypertension, and it's moderately effective for a very severe disease. It's an endothelin receptor antagonist. Preclinically, it was very clean. No toxicity in animal models. It all looked good. Turned out when it was put into people, and this was after the development occurred, it turned out that about 10 to 15% of people develop cholestatic liver toxicity that brings them to clinical attention. Because of that, the drug received a black box warning and was a real problem. And again, it was not predicted at all by what occurred in the animals. And one thing to be aware of is it's an inhibitor of BSEP. For those of you who know what it is, it's involved in bile acid transport. But the BSEP inhibition does not explain the human-rodent differences. It's as potent a BSEP inhibitor in both of the species. So what we did was we just gave our humanized mice. You can see that if you give a control mouse bosantin, absolutely nothing happens. But when we gave our humanized mice, within 1 week, the ALT came up and stayed up, so there was clear evidence of drug toxicity. We could confirm it with a variety of other measures. The alkaline phosphatase was up, the GGT, and a whole bunch of other things that you could look at. And again, it was dose dependent and so on. So here's a second case where you could identify a human-specific toxicity that would not be detected in rodents. And I could show a few other examples with other drugs, but I thought I'd just say that we've gotten a little more sophisticated with our studies, and we've done basically two things with them. We've done in situ perfusion. Some of you are probably familiar with that. And we can compare and look at differences in clearance and metabolism and so on by using this kind of a method, just comparing the humanized liver to the control liver. And we can tell you if there's going to be species-specific clearance differences. And the other thing we do is, this is our assembly line here. These are metabolic cages where you put the mouse in, and over 8 hours, 24 hours, you can collect urine, feces, et cetera, and do complete disposition studies. I won't bore you with the data, but I'll just show you one thing. This is cefmetozole, a cephalosporin antibiotic that's of interest because it's known that it's cleared very differently in rodents and in humans. And what's interesting is if you give a control mouse cefmetozole using that system I just showed you, most of it is cleared through the biliary tract and comes out in the feces. In contrast, if you get the humanized mouse, most of it is cleared renally, which is exactly what you see in people. So the clearance mechanism in the humanized mouse indeed predicts what's going to happen in human. And I won't spend a lot of time on this, but by using that in situ perfusion system, you can measure uptake, binding, et cetera, and we can explain the species-specific differences with some very nice parameters that explain why the rodents, the drug is cleared one way and humans another way. So it's a nice example of how you can predict differences in clearance and know what to look for. But let me just show you one last toxicity thing, because it's one I really like. And we did a thought experiment with Lasix, or furosemide. Lasix is a drug that's been used for over 40 years. Low dose, you know, it's non-toxic. It's used for treating edematous states. However, more recently, it's become clear that high-dose Lasix can be used in acute situations with severe heart failure. And you give doses anywhere from 250 milligrams up to a gram, pretty much IV push. And it's a very effective way of treating acute heart failure. However, there was an interesting thing. If you give a mouse a 200 milligram per kilogram dose of Lasix, which allometrically is the equivalent of a 1 gram dose in human, a funny thing happens. You get very severe liver toxicity, hepatocellular necrosis. Now, if you were trying to develop Lasix as a treatment for acute heart failure, this could easily be a showstopper. You would say it's going to be toxic in humans. So the question is, could using the humanized mice give you key information that would enable a drug that would be safe and effective in humans not to be barred from development because of a rodent toxicity? Any of you who've been around industry will be aware that that is not an uncommon problem. So what we did was we gave control mice a high dose of Lasix, 200 mg per kg. This is the ALT level. 1 day later, it's way up. But you'll notice the humanized mice, you give them the same dose, there's no evidence of toxicity based on serum enzymes. And if you look at the histology, it's quite nice. Right here, there's necrosis right there in the typical position. But in the humanized mouse, there is absolutely no evidence of necrosis. You see no toxicity. There's no enzymatic activity of indicators of toxicity. And what's interesting is I chose an area to show in here where there are some residual mouse cells in there. The eosinophilic in the size is different from the human. And what's interesting is the presence of the human cells even protected the mouse cells from the toxicity. Why is that? Well, it turns out Lasix gets converted. This is a furan. You open up the ring, and it generates an epoxide, which goes and binds to proteins and causes the toxicity. And you can't measure the epoxide because it's a transient intermediate, but it gets converted into this carboxylic acid, a KCA or ketocarboxylic acid. And what we did was to measure the amount of this indicator for the epoxide formation in the liver of a control mouse here, and then in a humanized mouse, and it was about 50-fold higher when it was in the liver of the control mouse. And so what's going on is the mouse is able to metabolize the drug more efficiently into this toxic intermediate. So besides showing that it's safe in humans, you can also look at the mechanism for the rodent-specific toxicity. So we were quite happy with that. And we're now in the process, we're in contact with the NCTR, which is the research arm of the FDA, and we're looking at a few more drugs that they've asked us to look at in our mice. And we're hoping at some point to create a phase 0, where you actually use these things right before you go in the clinic to do the testing, and I think that will improve drug safety. But rather than continue on down that path, let me talk about some more recent things we've been doing with the mice that I think relate to personalized medicine. We've been trying to model human genetic liver diseases in these mice. And the concept is we could take fibroblasts, make iPS cells, induce the iPS cells to become hepatocytes, inject the hepatocytes, and see if we can recreate the human disease in our humanized mice. And for today, I'll talk about Alagille syndrome. Some of you may have heard of it, but it's an autosomal dominant disease. In other words, you need one copy of it. And basically, you make a defective enzyme in a defective protein in the Notch signaling pathway, JAGID1. And basically, every first-year resident in pathology knows about Alagille syndrome because if you look in the liver of a young child, usually age less than 5, who presents with liver disease, it's Alagille disease if the bile ducts are absent until you can prove something else is going on. So a typical thing with Alagille disease is your bile ducts are pretty much sclerotic or absent, and that's the characteristic of it. So what we did was to make iPS cells from patients with Alagille disease and compare the liver we got from control to those with Alagille. And the results were really quite interesting. Both of the iPS cells could reconstitute human liver, as shown by staining with human albumin. But where it was really interesting was when you looked at certain antigens, and I don't want to belabor it, but ZO1 is the antigen you need to form the bile ducts, the ductules. It's the thing that allows the cells to adhere. And then some of you may be familiar with MRP2. That's the transporter that moved cefmetozole. And if you look, control cells have plenty of ZO1, plenty of MRP2, but the Alagille, we really can't find very much of ZO1 or MRP2. And what's interesting is they make comparable amounts of albumin, alpha-1 antitrypsin, and all the other things, but anything that's related to bile ductules and bile duct formation is impaired. So we're recreating the human disease in these mice, and we were going to pursue that, but something funny happened on the way to doing that study. The postdoctoral fellow, Yuan Guan, who did all this work, he was taking a long time to move the iPS cells towards hepatocytes, and I kept asking him, where's the liver? Show me the liver. And he says, I'm working on it. So one day he called me over, and he'd been modifying our pathway for making human liver, the hepatocytes, in a couple of ways, which I won't go into here, but you're basically serially altering the growth factors. And he called me over to look in the dish, and I looked in, and there was liver there. He was actually making liver lobules, sheets of hepatocytes. They had lumen, and you'll see the lumen are surrounded by cholangiocytes. And we call them three-dimensional human liver organoids. And what's really interesting, and I'll show you, is we go from the iPS cell to something that looks like endoderm. They form these balls. It then evaginates into something that looks like a foregut structure, and then it goes to a hepatoblast and then into the full organoid with the lumen, hepatocytes, and cholangiocytes. So I'll just show you a bit of what it looks like. Maybe it's just easier. Instead of having to get cells from patients, we decided we would create our own disease, and we use something called CRISPR methodology, which is a way to go in and specifically engineer the genome of the control cells and introduce the mutation we wanted. The patient we used had a particular mutation in JAG1, and so he made, you can see up here, the control cells. They express albumin, and CK7 is the cholangiocytes. Maybe it's better seen down here. You see how the cholangiocytes, the red ones, surround the lumina, and then you have sheets of the green, the hepatocytes. Here's the Alagille ones where we introduce the mutation. You only have red, you have no green, okay? You don't make cholangiocytes, and you can see the morphology is quite different between the control and the Alagille guy. Now, why does that matter? Well, when you look in the wells, if you look in the control wells, you see things like this. They form nice little compact balls of cells with this compact structure. When we looked in the Alagille wells, there were these just large fluid-filled cysts. And what you can do is quantitate that. Control 1 and Control 2 are two different people, different genetic backgrounds, but they have intact intact JAG1, and then C1 mu and C2 mu have the Alagille change. And if you look, the vast majority of cells in C1 go to form these organoids. However, when you have Alagille, the mutated JAG1, you end up with 90+% of your cells going to these fluid-filled cysts. So in these organoid cultures, we can actually reproduce what's probably going on with an Alagille liver in development. And I had mentioned before that it recapitulates the steps in liver development. I won't go through it, but there's very characteristic stages. And the key thing is you make hepatoblasts that surround the central vein, and then they differentiate into the cholangiocytes and the hepatocytes. And what Yuan did was to he stained mouse embryos in the liver and then stained at various days the organoids that we make. And it's really beautiful in that you can see the antigens coming up at different times as these guys are differentiating, and it's basically recreating embryonic liver development. I'm not sure, maybe there's time. You want to see a good movie? It's 2 minutes. Basically, Yuan's kind of a little bit of a Spielberg, and you're looking through a confocal microscope going through different depths in the field, and then you're looking at different days. So here now your foregut, and this is a foregut antigen, and so on. He takes you from the endoderm stage to the foregut, and it goes on, but I think I'll just show that. But the idea is these things are now recreating liver development. So can we actually learn something from this about the disease? With Alagille, you can't go in and serially biopsy and study it, but with 3D LOs, you can. So what he did was to take RNA from the various stages, endoderm, foregut, hepatoblast, and then finally when you get the differentiated organoids, and we've looked at a lot of different mRNAs, but the key one is JAG1. And what's really cool is JAG1 peaks at the hepatoblast stage, which is right when you're making the decision, should I differentiate? And then it goes back down. In the Alagille subjects, JAG1 is always lower, which is expected, but what's interesting is it's never regulated. So basically, Alagille syndrome results from the loss of the dynamic regulation of JAG1 expression, which is really cool. And just to show you the manifestation of it, this is day 20. This is a typical lobule. You can kind of see the lumen. They're surrounded by CK7 positive cholangiocytes. But if you look at an ALGS organoid, you'll note that there's green, meaning the hepatocytes, but there's not red. There's no cholangiocytes. So it's quite amazing, and you can see the difference. I don't know if you can appreciate if the lights are nice, but you can see JAG1 in the control, but you can't see it in the Alagille. So it's really kind of neat that now we can probe mechanisms in genetic liver disease, and you can put together a model. In this case, I think you've kind of got the idea that it's a loss of dynamic JAG1 expression, which is what creates the phenotype that we see in Alagille. So I hope I've shown you that mice can impact a number of areas: predictive pharmacology, the toxicology. I do think the personalized medicine and drug testing was exemplified by doing the iPS cells, and I think not too distant future, some personalized mice may be coming to a lab near you in which you can test therapies and pharmacogenetics and so on. But maybe just to finish off, I just want to show you how we're getting it back towards the clinic, which is a new way to treat liver failure. Right now, the only thing that can be done is orthotopic liver transplantation. Probably a few of you have been in on those surgeries. There's a lot of morbidity, a lot of costs, and a lot of problems with it. But what we hope to do in the not too distant future is change that paradigm. Could we do a liposuction, pull out stem cells called adipocyte stem cells from the fat, convert the fat cells to liver, and right now we can do it in 9 days, and then put those cells back into the liver. The advantages are you're just doing it percutaneously. You don't need immunosuppression since everything's autologous, and it would change things a lot. So can we do it? There was a Japanese group, Dr. Ochiya, that developed a method for converting these adipocyte stem cells into induced hepatocytes. We call them chemically induced hepatocytes, or iHeps. The problem is it takes about a month. It's about 2 weeks to get the adipocyte stem cells and 2 weeks to induce it. And the efficiency was incredibly low, below 1%. So Dan Zhu, shown here, worked on that problem, and now we can convert from the lipoaspirate to the, we call them spherical cell-induced iHeps in 9 days, and the efficiency, as you'll see, is close to 90%. So just to give you an idea, if you did the old style things, you were about $100,000 per liter in terms of growth factors to do it. With our method, because it's fewer days and it's more compact, we're now down to less than $2,000, and that's for GMP level reagents. Can we do it? Do they work? So the idea is we will gancyclovir treat and inject the ISAP into our mice and then see if we grow a human liver. The only difference is we bought a very fancy ultrasound machine, and we're going to do it percutaneously in the mouse under ultrasound guidance. This is the needle going into the liver, and we can do spot injections in the mouse liver. And just to show you what it looks like, so we inject ISAP into the mouse. This is the amount of human albumin. These are 3 mice. They continuously make albumin. And if you go in and pull out and do the human immunostaining, we're getting right in the areas we injected nodules of human liver. And they do proliferate. I won't spend too much time on that. But the key thing, the key problem with a lot of the IPS methods for regeneration is tumorigenicity. So for example, we put the ISAP under the kidney capsule of these immunocompromised mice, waited a month, and there were no tumors formed. In contrast, if you put our iPS-derived hepatocytes under the kidney capsule, within 2 weeks, you can feel the tumors formed. And when you look at the histology, you know they're true teratomas because they have all 3 germ cell layers there. So with adipocyte stem cell-derived material, we don't think the tumorigenicity is at all going to be a problem. So the bigger question is, can we do it? Can we scale it to make this feasible in human? The liver is your largest internal organ. It's pretty massive. And we're going from a 1.8 gram to a 1,500 gram organ, from a 1.8 in a mouse to 1,500. And we've calculated the number of cells we need given our regenerative capacity we've observed, and we need somewhere around 8 times 10 to the 8th cells to do it. And we want to get it from 1 liter of liposuction. Unfortunately, in America, getting 1 liter of lipoaspirate is not a problem from a person. So we've also scaled it up. We were doing little hanging drops to make our ISAP. We now call them SF-Heps for spinner flask Heps, where we grow them in these stir bar chambers. And just to show you, from half a liter of lipoaspirate, our last couple runs, we've gotten 2.2 times 10 to the 9th cells out of it. So I think we're on track now numerically. They, in terms of percents and so on, is shown here, but this is really nice. This is the amount of albumin they make, normalized on a per cell basis. Our SF-Hep cells, at least in terms of albumin production, seem to be very happy. So where we are is we've met with the FDA, and we've defined what are the steps, what quality control do they want, microbiology testing, functional characterization, and so on. And we think we can then perhaps make it possible for us to do this thing in the not too distant future, where you go from a lipoaspirate, pull out the stem cells, convert them to hepatocytes, and then put them back in. So that's kind of where we're at on that. And just to finish up, so I've shown you the other areas, and now we can add regenerative medicine to the list where these humanized mice have had some impact. And I do want to acknowledge, it's very important, we often don't thank our funders enough, and so I want to acknowledge our funders. Well, no, these people actually weren't our funders, but they're the people who should be funding this stuff. Some of you may know this is the most interesting man in the world. I'm a big fan of his. This is a CME course, right? This is CME. So your CME question is, what religion is the most interesting man in the world? He's a Jewish actor from New York. He's not Mexican, just to let you know that. So these are our actual funders, and I'm happy to stop here and answer any questions.
Thank you so much for this fascinating presentation. Any questions? I think I'm going to start with some questions. So I have two questions, actually. Is there any other humanized organ which has been, you are aware of, like humanized kidney in a mice or something like that?
So you're asking, could you pull this approach for other organs?
Yeah.
Okay, I chose liver for a very, very specific reason, and that is that the liver is inherently regenerative. You probably know that if you slice off a bit of the liver, it can regrow. So in that sense, the liver is optimal for trying to do this. The other advantage is you can survive for a certain amount of time with impaired liver function, which is when we were optimizing our transplantation regimen, we had to give enough ganciclovir so the liver was substantially damaged, but the mouse could survive long enough so the human cells could take over. I'm not sure how we could do that with the kidney. I guess we put the mouse on dialysis or do something else.
So
but
the reason actually I'm asking this question is that, which is related to the presentation, is that, for example, testing the toxicology of a new drugs in animal model is not really representative of what's exactly going to happen in human. And the question here, why are we doing that? And how much you know of data available which relate to toxicity in animal, which translated to toxicity in humans?
Yeah.
It's not always constant.
It's a huge problem. Much of the actual data is buried in pharmaceutical companies because what happens is the drug moves into the clinic, something bad happens, and it never sees the light of day. But the numbers I was told is somewhere around 40% of the toxicities that appear in clinical trials that are unexpected were not predicted at all by the animal toxicities. Because what you may not know is if you've ever been involved in drug development, it's all about a therapeutic window. What's the toxicity ratio to the efficacy ratio? And you decide a certain size window is off. Sometimes your window calculations can be a little bit misleading, but almost half the time there was absolutely no indication whatsoever from the animal data as to what was going to happen in humans. So that's the problem.
Any questions?
Gary, great science. Is it you've chosen to damage the mice liver, then to put in factors to convert it into human liver tissue? If you were to do the same thing to stem cells, would you get the same result?
The answer is yes. We took adipocyte stem cells, pushed them to become liver, and got that to work. The adipocyte tissue, remember, is mesodermal in origin. We've also been able to do it now with iPS cells and induce them to become liver. There's a big area in the field of what's called transdifferentiation, which may be a little more of what you're asking. Could you just put certain transcription factors in and convert the stem cells into liver or into the other things? And it's actually an interesting area. In the mouse, there are certain transcription factors. There's 3 of them you can put in, and you can convert a fibroblast into a liver cell. Unfortunately, it's a lot more complicated in the human, and people are just now starting to work out how to do that. But my bias is I really like the adipocyte stem cell because it's a very versatile cell, and you can get a lot of them. I mean, we're now making 10 to the 9th of them. So I think that may be a better way than to try the transdifferentiation, but we'll see what works out in the near future.
Another question. Can I ask you another quick question? What is the mechanism by which a humanized rat, because that's what you've got, changes its metabolic pathway. What are the control factors that do that?
In terms of drug metabolism?
Yeah.
Oh, okay, yeah.
You know, how does it switch from from sending a drug through the new liver when it's previously gone out through the gut or through the kidney?
Yeah, good question. Remember, in our mice, the only thing that's humanized is the liver. So if there is extraintestinal, extrahepatic metabolism, all that is still going on. The rest of the thing is still mouse, so you have to be careful with that. But we are working on inactivating the phase 1 enzymes in the mouse, where we can eliminate the mouse metabolism and really increase the human. Because the important thing to note is the rate of metabolism by mouse hepatocytes is 5 to 10-fold that of human. That's just an inherent difference. So even if you have a small residual of mouse cells, 5% or so, you still can end up with a fairly significant amount of murine metabolites. And so our next generation mouse will deal with that. Thank you.